We know what happens now when you have a single base problem in DNA, whether it be an addition, an elimination, or whether it be a substitution. We know that we can have mutations within our genetic code in a simple way like that, but what happens on a larger scale when we have genetic mutations that are affecting entire chromosomes? That's what we're talking about today in terms of aneuploidy. You can see in this image, these chromosomes, we don't have homologous pairs for all of them. That A3 there only being a single chromosome and B7 having three. These sorts of issues are known as aneuploidy, and the second type of mutation we're going to be looking at. So these are genetic mutations that are occurring on a larger scale. They're not affecting just a single nucleotide. They're affecting generally whole chromosomes or large parts of them, and they're affecting multiple genes at once. So the result of this is not going to be just a single change, say in maybe a section of pigment on your skin, but we're looking at whole body systemic changes, and they're going to have significant impacts. These types of mutations, they're generally occurring during mitosis or in meiosis when we're making our gametes, and that's how these changes can be passed on to offspring. So aneuploidy is what we're talking about. And aneuploidy is when we have an incorrect or abnormal number of chromosomes due to loss or gain of one or more chromosomes. So we know a normal eukaryotic cell is diploid, 2N, or our gametes are N, haploid, meaning that they have 23 chromosomes. When we're looking at an aneuploid cell, we're generally looking at N plus 1 or N minus 1 or N 2N plus 1 or 2N minus 1. So these are our aneuploid or abnormal chromosome number cells. And this is generally the result of, as you can see in the image there, non-disjunction, failure to separate during meiosis. So non-disjunction occurs when either a pair of homologous chromosomes, if we're talking about in meiosis 1, or sister chromatids, if we're talking about meiosis 2 or mitosis, fails to separate completely. What that means is we have two cells that result from that cell division, which have unequal and abnormal chromosome numbers. So if we have a look at the diagram here in meiosis 1, we see non-disjunction of our homologous chromosomes, which results in 3 chromosomes going to our top cells there and only 1 going to the bottom cell. The result of this, once we then go through meiosis 2 and produce our gametes, is that we end up with 2 gamete cells that are N plus 1. So they have a normal N number, haploid number of chromosomes plus another one. And then we've got our bottom cells there end up producing gametes that are N minus 1. So they have 1 less than usual. So this is aneuploidy, or they are aneuploid cells because they have an incorrect chromosome number. And it occurs as a result of non-disjunction, a failure to separate during these cell division and nuclear replication processes. You can see it represented here. We can see non-disjunction of our homologous chromosomes in that first diagram occurring during meiosis 1. And then we can also see it occurring during the separation of the sister chromatids, a non-disjunction of sister chromatids in meiosis 2. Now, if you have non-disjunction occurring in meiosis 1, then we will produce 4 aneuploid gametes. Whereas if it occurs in meiosis 2, we will still have 2 euploid, which means normal gametes, and 2 aneuploid gametes. So you should remember, means true from Old Latin, and an means not. So aneuploid means not the true or not the correct number of chromosomes. So what does this mean at the end of the day? Well, we end up with N plus 1 being known as trisomy, tri meaning 3, somy meaning chromosomes. So they have 2N plus 1, okay, so they end up with 3 chromosomes. Hence, trisomy If we end up with 2n minus 1, that's our monosomy. So they have one less. And these are our most common forms of aneuploidy. So if a zygote has 3 of a particular chromosome, whatever that chromosome might be, could be chromosome 3, could be 18, then that is known as trisomy. But if it only ends up with 1 copy, then that's known as monosomy. So these types of mutations types of mutations that occur during mitosis result in mutated cells, which can cause cancer, other diseases, and stuff like that, and won't be inherited. So if we have this trisomy and monosomy happening during mitosis, then that's going to cause a local mutation for the person. If we're looking at this occurring in gametes, that's when it's going to cause massive problems for the offspring. We can always see these examples of aneuploidy in a karyotype. So if we look at this karyotype here of these individuals, okay, looking at the first karyotype there, we can see that they've got 22 somatic chromosomes and the 2 X sex chromosomes, and they've got 2 of each of those chromosomes, 2 of each of the 23. So that means they are euploid. They have the normal number of chromosomes. If we look at the second example, this person is aneuploid for chromosome 21. So if you have a look down the bottom there, they have 3 copies of chromosome 21. 3 copies of chromosome 21, known as trisomy 21, or probably better known as Down syndrome. It, I would say, is the most widely known example of aneuploidy. When we have aneuploid zygotes existing, so we've had an aneuploid gamete that's been fertilized and formed into a zygote, a lot of these will end as miscarriages. So the body will recognize that these are not normal or typical cells, and as a result, they will be miscarried. But some of them will survive, and some of these examples of aneuploidy will be, fairly normal functioning people. Down syndrome, as I said, is probably the most well-known. Trisomy of the 21st chromosome, so trisomy 21, as it's often known. And it's when we have aneuploidy of our non-sex chromosomes, because 21 is one of our somatics. There are a huge variety of different symptoms we can see here, and they do vary from person to person with Down syndrome. But delayed mental development, physical malformations can also be an example, and probably the most obvious ones. But there's also some really positive symptoms of Down syndrome as well, where Down syndrome people are generally quite affectionate. They're very cheery, very positive, very kind people. So, there's positives and negatives. When we look at non-disjunction occurring in our sex chromosome, that occurs in some syndromes that you may not have heard of before, and it can result in some abnormalities. And it's important to note that these examples of aneuploidy sometimes can be minor in terms of symptoms and often won't get picked up, or picked up in only some cases. So an example is Klinefelter syndrome, which is where we have XXY. So we have the presence of an extra sex chromosome, and occurs in 2 out of 1,000 men. The result is that the person comes across as phenotypically male, though they tend to struggle in terms of developing masculine traits. And we'll look at some examples of that on the next slide. Turner syndrome is a monosomy, whereas Klinefelter is a trisomy. So Turner syndrome is the absence of a sex chromosome, which means that the individual only has a single X. A single Y will not survive. These people will not be born. It affects 0.4 out of every 1,000 girls. They have an XO genotype, and they appear female, but generally with some abnormalities. Primarily, they show short stature and delayed sexual development. So they look quite childlike for long periods of their life. So you can see here, these are 2 examples, Klinefelter and Turner syndrome. So people with Klinefelter syndrome, XXY, they tend to be taller, They tend to fail to develop significant body hair in terms of beard growth. They don't get chest hair. They tend to have a more female pubic hair pattern, which I don't really know what it means. They will have small, underdeveloped testicles, and usually a slightly more sort of, suppose, stereotypically female physique with breast development and less sort of broad shoulders and a larger upper body. In Turner syndrome, XO, you can see there, as I said, they do tend to show sort of a lack of female sexual development. So they do look quite childlike for long periods of time and into their adult life. One thing that can occur with Turner syndrome is a lot of these symptoms can be offset by hormone replacement therapy. So there are a couple of examples there of our aneuploidy in terms of sex chromosomes. When we're looking at aneuploidy with sex chromosomes, Klinefelter and Turner are potentially some of the more common ones, but it's important to note that we can have a lot of examples of aneuploidy in terms of sex chromosomes. So we can have XXX as well as XXY. We can have XXXX where there's 4. So there's lots of forms of aneuploidy in terms of sex chromosomes, but they are significantly less common. The thing with these aneuploidies of sex chromosomes is that they don't tend to result in miscarriage like our other aneuploidies, because a lot of the general bodily function is controlled by the chromosomes that aren't having any issues with aneuploidy. Last thing to go over is looking at the inheritance of these mutations. So it's important to know that any mutation that occurs within a somatic or body cell cannot be passed on. So if I get skin cancer, that's a mutation in my skin cells. I can't pass it on to my children because that occurs in a somatic cell. The only genes, the only genetic code that I'm passing on to my children comes from my gametes, which are the result of gametogenesis, meiosis, occurring within testes or within whatever gonads a person tends to have. So mutations within somatic cells can be passed on to future daughter cells, but can never be inherited by the offspring of that person. Cancer being a prime example. You can see in the diagram here, we have a mutation occur in our 3rd generation there, and that gets passed on to all daughter cells coming after that, but it won't be passed on to any other cells or any other offspring. In order to have a mutation that is passed on from generation to generation, it needs to occur in our germline cells. So that's the cells that gametes are produced from in our gonads. If there's a mutation that occurs within the germline cells, then that can be inherited from the next generation because those mutations will be incorporated into the gametes themselves and then into every cell of the offspring. So mostly these mutations will result in what we call congenital defects and disorders. So that's disorders or defects that you're born with. You can occasionally have mutations that will be advantageous. So mutations aren't always a bad thing. We'll look at this in more detail in evolution, but you could have a mutation that makes you better at functioning, gives you an advantage. A great example of this is Michael Phelps, widely regarded as the best swimmer to have ever lived. I think he has the award for most gold medals ever won by an individual. And he actually has a mutation that results in a greater number of mitochondria in each of his cells. And that means that he can make better use of any available oxygen, glucose, and energy and produce more energy, which makes him a superior athlete. So that's an example of an advantageous mutation. In terms of variations and looking at the variations that result from these mutations. So the mutations of genes in germline cells creates new alleles and creates new phenotypes. And this variation is one of the things that we base the entirety of evolution on, that variations exist and that natural selection can select for or against these variations. We'll look at this in more detail when we do evolution. For now, that's it for mutation.